obvious characteristic of the Asian citrus fruit. So if you spot any kind of this bug on your tree, then you need to be very careful about it could be the Asian citrus psyllids. And these yellow little bugs are the nymphs, and they will produce the wax tubular in here. And so this is a picture from one of our testing in Florida's orchids, and I have a pretty high acidic pressure over there. So once the tree be infected, this is a typical symptom, blotch pattern on the leaves, and it's very easy to confuse with the nutrition deficiency. And once the tree being infected, you will have dieback symptom. Eventually, it will die, and it can kill a tree as short as within like 5 to 7 years. And this is the symptom of fruit. So when the plant got infected, the trees will produce those fruit. They will lose juice, drying, got bitter, so it's impact the yield of the fruit a lot. So other than the Silas, another candidate is Laterbacter patera that also infect other plants, like the potato plants, the Solanaceae plant, including potato, tomatoes, and it's transmitted by the potato psyllid. So the disease petal system is a little bit pretty similar to the Asian citrus psyllid and the citrus greening disease. So in our research, we are using the both system then to try to find the solutions. And for citrus greening, the pathogen, they can infect the plant, not only on citrus. Basically, all the trees that the Asian citrus psyllid can feed out, they kind of can infect it by the Silas bacteria. So including almost all the variety of citrus, kumquats, mandarin, grapefruit, orange, and they are hybrids. Other than citrus, those close relative to the citrus, including curry leaf, orange just means they are widely used as ornament plants in probably just like for the road trees or maybe next to your orchids. So be careful about those trees used for ornaments. Do not keep those too many around your orchids. They could be a host for the Asian citrus psyllids. And usually if you do the management, you can be skeptical because you think those are not your trees, right? And not the citrus trees, you should not be a problem. But they do, they will become a host, a sweet home for Asian citrus psyllids. So do watch out for those alternative hosts for the psyllids. So following that situation, we have an expert coming today. I believe Dr. Grosser will give us more update later on, and also give us promising updates, promising update of the research, what they are doing to manage the citrus greening disease. So that's the current situation, and I've been working on this disease for many years. Every year we try to check the situation, but I think so far from the yield, that's no good news. And in last year, we can see the production is come for, Florida have 90% reduction in the yield. So yeah, the situation is kind of desperate, not very good, but yeah, later on we're going to dig out what's the promising updates, research from Dr. Grosser. So yeah, Florida used to be the lead production state of the citrus. So, but now it's taken over by California, but their industry is very but challenging is there. When I was in California, it's kind of the situation. Sometimes people feel, yeah, our situation could maybe we are better than Florida. The weather we have probably silly are not favor this condition, but let's see the realities. Okay, this is a map I pulled out from 2019, and the first case being identified in Hacienda High here. So at the same year, on 2012, they removed all this is not a citrus production region, so it starts from a backyard trees. So the homeowner bring a branch to the grafting, and it's obviously a sea loss positive branch, and the tree got infected and be detected. Because after Florida's situation, California has a survey program to survey the citrus tree, and they identify this and they try to remove all the trees surrounding this location. And yeah, 2019, you can see 7 years after they removed the first tree and 100 years tree around that region. This is a detection map and a quarantine boundary of the disease for 2019. So as you see, positive trees, boundary area, and Riverside, that's where I was working with. And then 3 years later, you can see the boundary, it's expanding, and more trees being detected positive. Yeah, this is Hacienda High first tree being detected, and obviously they remove all the trees surrounding it, but you can see it's kind of spread out and even more fell apart. And this is the most update map, and obviously you can see the disease is spreading. Riverside here, Hacienda High probably here, and now these are all setting quarantine boundary. And I always got the email from time to time, yeah, we are expanding our boundary in Southern California. So this is the real situation. So yeah, I've heard now their goal is to kind of slow down the spreading. And for us, it's not a very good news. Okay, then I'm going to talk about a little bit about the Taiwan situation. I grew up in Taiwan. I have a bachelor degree in plant pathology department of National Taiwan University. And this is a very old disease, have long history. Started from 1951. Basically, it's a very dark year of citrus growers in Taiwan. And it's a small island, and that several years, roughly 2/3 of the nation's citrus orchards are lost due to the citrus greening disease. And in Taiwan, we love orange tangerines because of the pronunciation. It's very close to Lucky Wilderness. So, when I was child, even now, my father always do, during the Lunar New Year's time or some festival, if it's in the season, he always going to purchase lots of tangerines, and because of the shade, we will make it to a small pyramid. Yeah, that kind of simple, like represent like it will bring in love and wilderness. Yeah, and we have many kind of varieties on the markets. It's around 30 different kind of variety, including orange, tangerines, lemons, pomelos. Yeah, and this is one of the pretty popular on the markets, one kind of satsuma. A little bit different from the local satsuma, I believe, but it's kind of some hybrids of varieties. So over 60 years effort, but this is last century, and hopefully in our generation we can make the pen easier and go over this situation quicker. So over 60 years in enhancing and prevention the control of this disease. And it's kind of about 15 years ago, occasionally we will hear about, oh, that's a small outbreak in which orchid, which region, but so far in these 10 years, it's pretty stable. And what they do, basically, early stage, remove the infected plants, and also provide and replanting the healthy seedlings, rootstock, healthy trees, and they start to use tea grafting technique to obtain, to provide growers the silas or the other virus-free citrus buds to for the replant all the diseased trees. And also very important, you need to implanting the sufficient integrated pest control or pest management program in the orchids. And basically the most important thing is you need to spray before the psyllid population being built up. Once the population is too high, the effect is kind of pretty limited. And use the greening tolerant rootstock, and then they also established the HLB monitoring and reporting system to provide a rapid response to any outbreak of the disease. Basically, it's just to prevent the psyllid population being break up or outbreak of the certain location. Once there's a disease outbreak, it will become a spreading source for the disease. Okay. So currently, the major disease management, I would say, very important. Remove the infected trees, do not become a spreading source, and also prevent the introduction of the Asian citrus psyllid. And so, once you see it, you should immediately try to control or manage the bacteria, the psyllids with the insecticides to reduce the pathogen transmission. Since we still haven't really have a very to say that the effective cure for this disease yet, so these two managements is very important for the current situation. So the research about the citrus to manage this disease is of course major focus on Asian citrus psyllid control. Vector control is important because they are bringing the bacterial pathogens everywhere. And also the other focus, in fact, tree therapies or cure. And we are researcher put lots of efforts also working on the technology to protect the new planting trees because younger trees can be more susceptible to the disease. And of course, the early detection technologies, if we can detect the disease as early as possible, we can prevent it to become a pathogen resource. And yeah, management practice to maintain the productivity and also generate HLB tolerant citrus varieties. And currently we have some possible disease management. Later on we will hear more about this part in today's symposium. And yeah, that's some antibiotics like tetracycline they can use to kill the bacteria and which seems to have significant health environmental, but which can bring some significant health and environmental side effects. And that's also lots of research are trying to use antimicrobial peptide to suppress the CLS. And the good things compared to the antibiotics, the good thing of the peptides is some of them can be used for external application, and some, most of them can be internal synthesized in the plant. So once we introduce the peptide into a plant, they can produce them automatically. You don't really need to do the external applications. And some biocontrol for the silice and the bacteria, and of course develop the the CLAS or the cilic resistant citrus cultivars. And here I'm going to briefly go through what we've done in the past several years in the research with Hailing. We start with a variety US 942. It's generated by Dr. Kim Bowman in UCA in Florida. And because this variety has 3 or more field tests showing a good resistance ability to HLB. And so we started the research from these varieties. And I'm not quite so sure if this variety is popular in Florida, Dr. Grocer?
942 is very popular.
Yeah, but from California, since they don't really like it. Yeah. But anyway, the soil condition, weather in Florida and California is very different. So yeah, they can also, I think it's also pretty obvious this disease is very localized because every location has their special soil, different soil type, weather conditions. So apparently only one variety is not enough, so we need to use other approach to generate more tolerant varieties. So yeah, we'll start with the US 942 and we compare to the susceptible varieties with the US 942 is a hybrid from the Cleopatra mandarin and the Panzyrus tribolata. Then we compare the US 942 to their parent line, the susceptible parent line, and we identify some positive regulators they can use to generate the resistant varieties. And another group in UCR, Dr. Ruth's lab, and his research scientists, they are using another totally different varieties to run a breeding program. So they are using the Australian fingerline to create the hybrids, to try to generate the resistant hybrids. So after we select the regulators, we further confirm their expressions in another totally different breeding sets of plants. Then we come out a set regulators we are with much more confidence they can be used for prevent this disease. So in the past several years, one peptide is something we are focusing on, and it's a novel peptide. And we found out it can directly kill the bacteria, including Xylella that cause the citrus greening, and Xyloso that causes Libra strip disease. And another, because these diseases are difficult to culture, these pathogens, bacteria are difficult to culture, and that's one species is easier than we can use medium to culture it. And we confirm it can also kill this species. And then we find out this peptide, the same peptide, can also induce the defense response to kind of make the plant not that susceptible to the disease, and also make the silics not preferring these plants. Okay, this is just some results. So this is the picture of germs, the CLAS, and the red color represent the dead bacteria, green color is the living one, and we incubate them with a peptide, and we are seeing the peptide that we test with different concentration of peptide, and also heat the peptide, and we can see the peptide can kill the bacteria. And even you heat it, the bacteria still preserve the activities and to lyse the bacteria. And we believe it's important because if we are delivering a product that can use in the hot weather in the field, the heat stable is pretty important for the molecular properties. And we have tested it on the diseased tree and also use it to protect the healthy trees. That means we inoculate the plant first and treat it with a peptide, or we treat the peptide and do the inoculation with the bacteria. And we have run multiple trials with different varieties of the citrus, and we all got pretty positive results that the peptide can kind of suppress the symptoms that the bacteria produce, and also prevent the infections. But these results are the ingredient in the greenhouse. We still need to see how it works in the field. So basically, until now, we have proved this peptide they have bactericidal activities, and they also can induce the plant defense response. And that's why they kind of make a plant that's not favored by the Asian citrus psyllids. So it can also suppress the Asian citrus psyllids growing on reproducing on the treated plants. And this peptide can be easily synthesized by the chemical approach and still have the activities, and it can also produced by bacteria and also produced by plant itself. And we have done some tests. We assume that they should be pretty safe for human consumption because it's you can find it in the fruit or some citrus related, such as the Australian finger limes, and it's being consumed by the human for a long history. Yeah, you may find it very common, but actually it's being used a lot in like making salad dressing or cocktails. Yeah, and we have done some tests. The peptide is sensitive to human stomach acid, so it can totally digest by our stomach proteins. And it's highly stable because it still keeps its activity under 60 minutes. Yeah, this is some data that's showing that the treatment plants that have is not that favorable for the Asian citrus psyllid. The plant have less insect bites, and the eggs and the leaves of the Asian citrus psyllid that have fewer productions in the treated tree. And we also found that the psyllids feeding on the peptide treated tree that kind of have less bacteria in the insect, and the leaves that produce on the trees kind of have acquired less CLS in their body. And now we are testing if we can introduce the peptide into the plant, that the plant to express itself. So yeah, because potato, they grow quicker, so we can quickly generate the transgenic potato to get the science from this. So here's showing two potato lines express the same peptide, and indeed they are more, they can produce more tuber and with lower bacterial titer, and because of this is called zebra chip, is because the bacteria in the tuber that will digest the starch to glucose, and when you fry it, the glucose will turn from much more quicker. So you can see here the transgenic potato, they are showing the frying chips, they are more similar to the healthy, non-infected tubers. So it kind of can indicate we can use the plants to produce the peptides, and it has the effect to suppress the disease. So, and also we also find that the potato psyllid much longer time to generate the citrus plant that can express the peptide. And now we are expressing the peptide in the chorizo, and it's a rootstock. Then we put on the scion on the transgenic, the transgenic chorizos. And this is just a signal. We can detect the peptide expression, and also in the scion part, that's not a transgenic plant. We also can detect the peptides in those scion tissue that graft on the rootstock. So that's a good sign for us, and now we're also using this peptide to treat other disease, including another vascular disease, red pear disease. Yeah, this is just a disease it looks like, and it will cause the leaf will lose the water. And basically, for the peptide-treated plant, we found the disease severity is not as severe compared to the non-treated plants. And also, we are seeing the lower bacterial titer in the peptide-treated tree. So, yeah, currently in my lab, we have ongoing project on comparing the citrus only, work with, still with UC Riverside and UF. Yeah, then now we are trying to use the citrus textura virus as a vector to deliver the peptide in the trees, and also use the intragenic approach. That means all the sequence we are putting in that can be through convention breeding programs to express this peptide to the important varieties to generate HLB resistant rootstock and scions. And yeah, we're also testing if the peptide can have any effects on suppressing CLS transmission. And yeah, what we can do here to prevent or slow the spreading of the disease, yeah, first, manage the vector, the psyllid is the most important thing. It's necessary to stop the spreading of this disease. And also check with your local LSU Ag Center county agent, I believe, and I'm here, yes, to consult of the insect management guide and need to be up to date. It need to be the up to date information because it will reveal the most updated situation in Louisiana currently. And yeah, and use the insecticide to control your vectors. And if you suspect your citrus tree are infected with the citrus greening, please contact our plant doctor, Roger Singh. Here is his contact information to report the case. We have the citrus fungal bean survey program running on in Louisiana. Okay. Yeah, last slide, acknowledgements about this work. Yeah, this is a group I work with in California, and yeah, now I'm here and setting up my own group, and we still heavily participate in the citrus fungal bean research, trying to find a solution for the disease. Then I'll stop here, and thank you so much today. And I'm here to learning about the situation here. So